And I'm going to start letting people in. Hey everyone, thanks for joining. We're going to be starting in a couple of minutes.
I assumed meeting. Yep, I wasn't sure. Yeah. Okay, that's what I thought. Okay. Like it'd be nice to do. Yeah, because I just submitted the amendment yesterday. So I haven't heard. Yeah.
Hey everyone, I think we can get started now. My name is Leah and I'm Bay Nature's events coordinator. It's nice to see so many of you today at the talk. Thank you all for making time on your Wednesday lunchtime hour to join. And I'm just going to give a brief introduction to Bay Nature for those of you who don't know, as well as to our events program before our talk today. So many of you know Bay Nature as a quarterly print magazine that comes out once every 3 months, seasonally, all about nature and conservation topics local to the Bay Area. We also have a website that features weekly online news content, also about nature in our local area. But we're also, in the last several months, we've been working on building up our events program with events like this one, which include virtual presentations with experts, as well as in-person outings with experts that are all largely inspired by pieces that are published in our magazine. So this is an example of this one. This was a piece written by our former Bay Nature Fellow, Guananí Gómez-Van Cortright, and just really excited to be working with to be speaking with some of the experts from that piece today. So before we begin, I just want to invite everyone who is attending to please turn your cameras on if you're able. We would love to see your faces if it's possible and just see you during the course of the presentation. So yeah, if you're in a place where you can turn your cameras on, please do that. We'd love to see you. It's just great to see faces when possible. And additionally, if you have any questions during this presentation, please enter them into the chat and we'll get to as many as possible at the end. I'll be calling on your names and you can go ahead and unmute and ask your questions at that time. So we're going to be saving questions for the end, but yeah, please enter them during the presentation into the chat. Yeah, so thank you so much. And without further ado, I would like to introduce our two speakers today. We're joined by two Cal Academy researchers. First, I'll introduce Riah Evin. Riah is a marine biologist with a specialization in kelp forest and California rocky reef habitats. Riah works to educate the public through creating and maintaining recreations of these habitats, as well as the breeding animals found there. And we're also joined by Elora López-Nandam. Elora is an evolutionary biologist with research interests in the ecology, evolution, and conservation of marine ecosystems. Elora has used genetics to address a wide range of topics in the ocean, from sea cucumber fisheries in Fiji to coral bleaching in American Samoa, to how mutations accumulate in coral colonies. So we're really lucky to be joined by both of you today. Super excited for your presentation, and thank you so much. I'll turn it over to you guys.
All right, great. Thanks so much for joining us. I'm going to get us started right off. What in the world is a sunflower star? Or as you see here, some many-armed monsters, sunflower starfish. Their Latin name is Pycnopodia helianthoides. You will probably hear myself and Elora refer to them as Pycnopodia. or pycnos because that Latin is a mouthful. They are the largest species of star found along our coastline. They can grow up to 1 meter, so over 3 feet from tip to tip. They weigh up to 14 pounds and they can grow up to 24 arms. And those arms are covered with over 15,000 tube feet. That lets them be incredibly fast. So they can move about 1 meter per minute, which is really, really fast for a sea star. So you and I might cover that in a single footstep, but if you're a small little snail or an urchin, these things can just cruise over to you at lightning speed. In addition to having really cool biology, they are critical in our ecosystems. They fill the role of keystone species by basically being generalists when it comes to eating. So they'll eat California mussels, they'll eat, very importantly, urchins, they'll eat any kind of gastropod or snail. They also like their cousins, so they'll eat bat stars or ochre stars. And that kind of predatory pressure makes openings in an ecosystem, right? So if they come over a mussel bed, they can clear a bunch of space out for kelp recruitment. They can eat a bunch of our herbivores, like our urchins, and they can make sure we have diversity within other star populations by eating bat stars or ochre stars or other stars along our coastline. Unfortunately, a lot of that predatory pressure was alleviated in 2013 when sea star wasting came to our coast. Sea star wasting manifests, you can see its progression on the right side of the screen here. It manifests as stars beginning to twist their arms into these really torturous yoga poses. They'll actually eventually start to tear their own arms off, or their arms can walk away from their body. And it is basically always lethal, and it's incredibly contagious. So this spread up and down our coastline. We lost about 99% of Pycnopodia in California, maybe a little bit more, and it had similar effects on related species and unrelated species. Basically, all stars in our waters were affected by sea star wasting disease. So why are Pycnopodia important? When we had Pycnopodia, this is what our kelp forests look like. Super happy, healthy, beautiful forests underwater. It's like walking into the redwoods when you dive into these. Without Pycnopodia, our urchins, our purple urchins and our red urchins, who are aggressive algae eaters, were able to come in and tear down these kelp. These kelp can grow up to 3 feet a day. They can easily reach up to 60 meters in length. They're massive, and these urchins can come forward and basically just eat them off at their holdfast, which is kind of like their root system, or they can actually tear down entire plants and eat the whole thing. So without Pycnopodia to keep that in check, this is what our kelp forests, a lot of them, 90% along our coastline, look like today. This is an urchin barren. So the urchins will eat all of those beautiful upper stories. They then eat the understory, like our pterogophora, some of our smaller kelp species. And in this picture, you can actually start seeing some of the underlying granite. So they're eating through even the calcareous algae, just the turf algae, the basic algae. You never saw this before wasting disease. Seeing granite underwater is mind-boggling to me. It's very scary. Luckily, we know what eats these, so we know Pycnopodia eat them. So the Academy started a program to start breeding these. We started with a literature review. We are looking at an endangered animal, so we're not just going to go crazy experimenting on them, right? We're going to do a literature review, see what people have done in the past, and then we're going to start studying related species in-house. So we have a lot of stars that live in the Academy. Start trying to spawn them, create effective and safe methods on related species, and see how we can rear them. So we started that project in 2022, learning how to spawn them and the food that they were going to need, right? So we began spawning experiments with our bat stars. That's what you see on the left. We gave them an injection of 1-methylaldenine or 1-MA. This is a biochemical cue that stars create in their own bodies. to stimulate spawning in the wild. And here we just gave them a little help, so a little injection of their biochemical cue. And we had great success and absolutely no disease, no mortality afterwards. So we proved that this could work. And we also needed to raise their food. So as much as I would like to be out on those urchin barrens, picking up urchins and feeding them to baby stars, I don't have time to do that. And we also need food that is of a specific size, right? So a baby Pycnopodia isn't going to be able to eat a full-grown purple urchin or a full-grown red urchin. But if we can feed them baby urchins, great. So we started that. Potassium chloride, similar cue for our urchins. It causes their ovaries or testes to contract, thus stimulate spawning. And again, wildly successful. No injuries, no mortalities in the animals that we worked with in-house. So we have their food. We have a really great ability and method for spawning them. We did not have a great way of rearing their larvae. So we had their food. Their larvae love phytoplankton, and luckily we have a great staff at the academy who's able to rear phytoplankton without an issue. What we needed to do was create water motion for their larvae. So actually, let me go back real quick. These are their larvae. So they look wildly different than the adults. They've got this bilateral symmetry. They've got these cool appendages growing off. They need to be up in the water column to eat that phytoplankton. So we needed to recreate water motion. Going back to that literature review, there are historically labs that have raised echinoderms in a lab setting that use something called a Strathman apparatus, which is just this giant, huge, basically motor-driven paddle. And we didn't have space. We had little tiny candy jars to do this in. But people had done this successfully in stagnant bins. So we figured we'd try that. It was easy. We could do it in small scale. We have also had, so the stagnant bins are here and here. This bin on the left, this is how the Academy has historically reared fish to great success. So this is a flow through. There's a very small trickle of water coming in, a very small size screen. So it'll keep the larvae in and still allow water turnover. And then there's an air stone in there to provide more agitation. So we wanted to try that since we knew it worked with fish. Maybe it'll work with echinoderms. And then we wanted to try kind of two off the wall ways of providing some water motion. So I snuck into our veterinary lab and snuck back out with a stir plate. So that's just a basic stir plate. It's got a magnetic stir bar. You'll see those in any lab. So we wanted to try that. And then also one of my colleagues, Nick Yim, mentioned that pot stirrers exist. I didn't know this was a thing. So in kitchens, you can use automatic pot stirrers to keep like your Alfredo from burning or something, right? So you don't have to sit there and stir it all the time. They're cheap, easy, can get them right off the shelf. And they've got a backup battery. So if my power goes down, I still have water agitation, which is great. So we set up this experiment. We had those two stagnant bins. Those were kind of our controls because the literature said this is what's going to work. We had our fish flow through, which we also thought would work because we've had huge success with it in the past. And then we had that magnetic stir bar and our stir mate, which is that automatic pot stir. So we bred our Proteria miniata. Those are our bat stars. Got lots of beautiful larvae, put them in this little tiny setup and saw what's going to be what's going to work for us. Unfortunately, our magnetic stir bar turned out to be completely lethal, so we learned a lot from that. Great way to experiment with a not endangered animal. Our flow through, so that fish rearing thing and our static bins were successful. It was great, right? We thought that these were going to work. We had between 10 and 20 animals settle out in those. So these do go through complete metamorphosis. The bat star larvae are over here on the left. They go through a complete metamorphosis into a star. So that's what settlement means. So we had animals settling out in there. And then the stir mate, I had over 300 animals settle. And I say over 300 because I stopped counting. We had so many animals settling out. Honestly, I thought I was going to get in a little bit of trouble with my boss because I said, maybe I'll have 50. But this was great. These are animals that can now go on exhibit. They can also be used as food. So we had an idea of what would work in a house, but of course we wanted to test this again, right? So we went ahead and tested it on a bunch of other things. So we repeated our bat star. We also, bat stars are Bacteria miniata, Strondril centauroides purpuratus, say that again. Those are our purple urchins. So we tested this again. It worked great with their food as well. And then our Dendrester eccentricus, those are our local sand dollars. So worked with these guys too. So hundreds of thousands of animals settling out. This was a tried and true and tested method. So we had a way of spawning our animals, of rearing their larvae. Now we were able to go to our colleagues and say, who wants to spawn some Pycnopodia? We did not have Pycnopodia in the house. So we went down to the Birch Aquarium on Valentine's Day of 2024. This greeted us down there. The Birch Aquarius had put together our little Cupid spawning area, which was really cute. We then injected our animals. This is a male. He took about 3 hours to spawn. So there was a lot of berry white blade. And we also injected our female. She was not spawning. And finally, at the 4-hour mark, we got eggs. So both of these the sperm was very, very motile. The eggs were super healthy. You see that nice peach color. That's what we want to see. We were able to put them together, and within less than 10 minutes, the sperm and egg had fused. We had our first successful California spawn of Pycnopodia. It was super, super successful, super exciting. There might have been some crying. There was definitely a lot of cheering. There was a lot of work that went into this moment. And then we brought them back to the academy. So my colleague Julie and I drove them all the way back up the 5 and set them up at the academy. And this is what our working lab looks like when it is full of larvae. So you can see all of our pot stirrers set up, lots of larvae. I may or may not have brought back 11 million larvae. It's fine. Everything's fine. So we were super, super successful, and then they settled out. So we were able to do a lot of studies and find out so much information from this first successful spawn. So one thing we found out is they have this really neat bimodal distribution with their settlement. So they start settling around 90-day post-fertilization, and then it kind of tapers off. And then we have this another huge peak right around 110 days. So that was really interesting and really good to know when we're raising these animals for the future. We were also able to do some experiments with density. So how many of the larvae can we pack into a volume of water? So we set up 4 experiments, 1 larvae per mil, 2 larvae per mil, 5 larvae per mil, and then what I kind of called our Hail Marys, because I did bring back 11 million larvae. Oops. We just put a bunch in a bucket. So between 15 and 20 million larvae, to see, if we can really pack these guys in, can we make a bunch for eventual release, right? We're going to be needing a lot of these in our waters since they're all gone. And there was a pretty wild distribution on this, not necessarily doing better at the higher numbers. What we did see was survivability. So we had a much higher survivability in those less dense experiments. So this is good to know for animal health and welfare, right? We don't want to have a bunch of larvae bumping into each other and we're beating each other up. So those less dense will probably be how we're going forward, which is a really neat experiment to do. Moving forward, this is one of our newly settled stars, super cute. Moving forward, we are analyzing some feed trials. So larvae are super easy to feed. Like I said, we can just pour some phytoplankton and they'll go find it, or it'll find them. But once they go through metamorphosis and they become those little stars like you just saw, we don't really know what we need to feed them. So we did some experiments. Spirorbis is a type of calcium dwelling worm. So they build little calcium huts and live in them. We thought that this would work because we see very young Pycnopodia living in eelgrass beds and surfing over the Spirorbis, and we thought maybe they're eating those. Urchins, of course, we want them to eat urchins and get those kelp barrens out, get our kelp forest back. Other stars. So Pycnopodia, we do know one thing when they settle, they are extremely cannibalistic. And when you're trying to rear a large population for potential outplanting, you don't want them eating their siblings, right? We want as many of them to make it through to adulthood as possible. So we reared bat stars and ochre stars in house, hoping that if we fed them their cousins, maybe we could convince them to eat those instead of their direct siblings. And then the kitchen sink was basically everything above that, plus anything that is in our commissary at the academy. So I was feeding them chopped up salmon, herring eggs, capelin, oysters, if you name it, and we had it, I fed it to them. We're also going over growth curves. So this entire time I was taking measurements of how quickly the larvae were developing, how quickly the newly settled stars were developing on those various feed diets. So we'll be analyzing those measurements and seeing how that affected them. And then I get to hand it over to Elora because she's doing some really cool stuff with genomics on these stars.
Great, thank you so much, Riah. So hi everyone, I'm Elora, and my background originally was in coral genetics. And so when I joined Cal Academy, I joined the Coral Regeneration Lab to learn some things about the genetics of the animals that we're raising in the aquarium. But when I was then approached by Steinhardt and asked if I wanted to also start working on the DNA of these Pycnopodia, I said, absolutely, yes, this is amazing. This is an incredible project. And really, it's a joy to be able to listen to what Riah does every day and really to see her work, her team. If she didn't emphasize it, I will emphasize it, that this is an incredibly taxing and labor-intensive project. And so the work that has gone into this is really remarkable by Riah and her team. So I get to look at the DNA of these aquarium-raised Pycnopodia. And the reason we care about the DNA of aquarium-raised stars is that when we're thinking in the context of, possible reintroduction into the ocean someday, we don't want to just put in a huge number of stars. We want to also aim to maximize the amount of genetic diversity that we're putting back out. Because genetic diversity is really the fuel upon which natural selection and other evolutionary forces can act. That's how you get a resilient population. But there's a lot of challenges in preserving genetic diversity in an aquarium setting. So you start with a small number of parents, and that can be challenging because if it's unmanaged, you can result in inbreeding. So breeding between closely related animals, even siblings, which can lead to detrimental effects down the line if that continues unchecked. You might also accidentally select for traits that are beneficial to the animals in the aquarium, but not so beneficial once they actually go back out into the ocean. And then, of course, there's all sorts of different random mortality events that happen either because it's part of the life cycle of these animals or because things happen, mistakes happen, etc. And so there's like all sorts of different bottleneck effects that can happen throughout the life cycle of these animals. And we've started to see that in the corals. So this is an example of a coral spawning at Cal Academy. And when we sequenced the DNA of a bunch of what you see in orange here are the names of a bunch of different coral offspring that were born in the lab. When we actually figured out who was related to whom and which parent came from whom, we found that 22 out of 23 of our surviving offspring came from the same two parents, and only one of them came from the other two parents. So we've started to see time and time again now that there can be a huge discrepancy in which parents are the most successful at producing surviving offspring, but we don't fully understand the reasons why. And so when we're thinking about this Pycnopodia project, we have this really cool opportunity to look at how genetic variation changes throughout development. So if you can imagine here that we have a jar of our Pycnopodia that Riah and team are raising, and that we track them throughout their development, this is kind of what it looks like. So this is day 1 of post-fertilization. This is a 1-day-old embryo. As they develop by day 13, they start to take on a more distinct shape. By day 23, they're a lot more complex. And then by day 35 is when you get to what Riah calls the noodle arm stage, where they have these really interesting appendages and they're able to actually hunt in the water column. And so even in the first 35 days of development, there's a huge number of transitions that these animals have to get through and have to be able to survive. And the reality is not all of them are going to survive these different stages. Not to mention the fact that once you get past, day 90 or day 100, they have to make the biggest transition of them all, which is this metamorphosis or settlement into that sea star body plan that we're used to seeing in the adults. And in addition, as Riah mentioned, our stars were also raised at different densities, and the success rate of settlement, so again, that's going from larval stage to this kind of juvenile star stage, larval densities that were lower had higher settlement rates. And so this is really interesting because my question here is, do you then see different genetic composition of the animals that were raised at different densities? And so this is the question that we tackled with this first cohort. So we took genetic samples of the population throughout development, starting on day 2 before they had been separated out into different treatments. And then again on day 19, we took a sample of the 1 larvae per mil and the 5 larvae per mil, did the same thing on day 33, and then took another sample once they were actually settled out. And so by doing this, we'll be able to track how variation differs throughout larval development and into settlement between 2 different rearing densities, so the 1 versus the 5 larvae per mil, and within a cohort of full siblings. So the thing that I'm not sure that we've mentioned yet is that for this first spawn that happened in 2024, we had just 1 father and just 1 mother that produced these 11 million plus offspring. And so this is a really interesting opportunity to look at how even within a cohort of full siblings, there can be a really interesting amount of genetic variation and differences depending on how you raise them. I'll also mention that we're using ochre stars as models for sunflower stars as well. So we've done a couple of different spawns of ochre stars that are held in-house at Cal Academy now. And we're doing very similar things of tracking their genetic development. And our veterinary team is also doing some really interesting studies to really improve and optimize the protocols for spawning these animals in the aquarium. And then, of course, we should also mention that this is an effort that goes beyond Cal Academy. So we are members of what's called the Pycnopodia Recovery Working Group, which is a really interdisciplinary set of partners across North America, from the US and Canada and Mexico. There is a QR code at the bottom of your screen if you're interested. There was this roadmap to recovery for the sunflower sea star that was published by the group led by the Nature Conservancy a few years ago, and it really lays out kind of a path to how we get back to being able to have more of these animals in our kelp forests. And then finally, I'll mention that we're also part of the very new Pacific Coast Ocean Restoration Initiative, which if you want to learn more about that, there's a QR code on the bottom of this slide as well. And that was established by a NOAA grant late last year to really address all sorts of different problems that are facing the Pacific coast of the United States. So it's kind of a 3-pronged approach. We are part of the Pycnopodia prong, but there's also groups working on abalone restoration and kelp restoration. And so this is a really interesting group to be a part of, to really try to work through the challenges of kelp forest lost from the lens of a couple of different organisms. So the future involves a lot more sunflower or Pycnopodia and ochre star spawns. We'll be continuing genomic characterization of these aquarium raised animals. We'll be continuing conversations with our partners about the path to reintroduction and, critical to that path to be reintroduction is the husbandry methods that Riah and team are developing. And then we're also going to be expanding efforts to find more adult sunflower stars that are living on the California coast. There's very few that are known currently. Every once in a while, you know, a diver or someone else will come across one, usually more in Northern California. I have really none in Southern California in the last many years. But we really want to understand who is still out there and who is still surviving and reproducing to maximize what we can do to help the ones that have survived. So I think we'll pause here and just thank the huge number of people who are involved in this work, as well as some of our funders who have made it possible. And you can see some of our tiny stars. They're much larger now. This video was from at least 6 months ago. Yeah, I would say the stars are more like,
yeah, they're like
this size.
they're a decent sized cookie.
Yeah. Yeah. But we're happy to take any questions that you all may have.
Awesome. Thank you so much, both you, Riah and Elora. And that was a awesome presentation. Super, super fun. And we have a lot of questions in the chat. So let me allow people to unmute first, and then let's see, and then we'll get to questions. So there we go. I'm just going to start from the top of the list of questions, and we'll go and see as many as we can get to. But yeah, if you have any questions, please enter them into the chat, and we'll get to as many as possible. So I'll just call out on people's names, and then if you don't mind unmuting and asking a question, or I'm also happy to read them out loud for you, we can go through the questions that way. So our first question, it looks like, is from Paula. Paula, do you mind unmuting and asking your question?
Yeah, hi. I'm Paula Shadle. I'm just really curious what's known about the wasting disease. Is it bacterial, viral, or environmental? Because when you go to return these Pycnopodia to the wild, they're just going to get sick, right, if it's infectious.
Yeah, that's correct. So there are colleagues of ours who are doing a lot of interesting work on the actual pathogen that causes sea star wasting disease. And they believe they have identified a cause. It's not fully public yet because it's kind of still under investigation, but we are much closer now than we ever have been to knowing what that pathogen is. And as soon as that is really fully determined, we'll be able to develop tests, you know, both in our aquarium systems and in ocean systems to be able to see when and where the pathogen is occurring. And then we're also in talks with people to really start thinking about how do you breed animals for resistance to that pathogen once it identified, because, you know, as you point out, there's not a lot of, you know, there are a lot of concerns about what does it mean to breed these things and then put them right back into the environment where their parents didn't survive.
Yeah, and keeping them going long-term in captivity sounds like really a challenge.
Exactly.
Thanks.
Thank you, Paula, for your question. We have a couple questions now from Josie. Josie, do you mind unmuting?
Yeah, hi, thanks so much. This is just fantastic to hear. I've been hearing little bits about the whole Sunflower Sea Star Lab and all your work, and it's very exciting. I did put a link. I'm going to be updating our Sunflower Sea Star page of our Mysterious World of Bull Kelp website, so to include all this information. I was wondering just the time frame of when that first spawn was from Birch Aquarium. Was that was a year ago from this past February?
Correct. Yeah, the spawn took place on February 14th of 2024. Yeah, Valentine's Day.
13 months.
24. Okay, cool. And then my other kind of question comes up when people say, oh, reintroducing these, you know, few sea stars that you are raising in the lab. As broadcast spawners, they need a certain density to have a successful wild spawn, right? So that seems just like a daunting idea of putting these loners out there And what is there some kind of idea of density that you need, or are you going to corral them to keep them in place to be close enough to each other to successfully breed out there in the wild? What are your thoughts on that?
Yeah, that's a great question. And we're working with our colleagues about how many we would have to release, because like you said, releasing 5 stars into the ocean isn't going to do anything. And these are extremely mobile animals, so trying to corral them, good luck. They can get through a lot. So it's kind of a question for all of us who are doing this work in labs. How many can we breed? You know, we're looking, and that cannibalism is a really big hurdle to get over. You know, how can we breed hundreds of these for release, maybe thousands? And at what size do we need to release them? You know, can we release them just after metamorphosis? Then that's easy. I could have given you 1,000, I could have given you probably 10,000
yeah.
stars last year. But if we need to keep them until they're a year old, they start eating each other right after metamorphosis. So that's a great question we need to answer.
And I've one other question is, I assume the same work is happening in a number of labs, right? Jason is continuing to raise his, is the Birch Aquarium also raising them? So you're all kind of sharing your data and info, is that?
Correct. Yeah, we're all working really closely together. So Jason, Oregon Coast Aquarium, Birch, Aquarium of the Pacific. I'm gonna miss some people. Sunflower Star Lab. I think Monterey is coming on board. Seattle Aquarium, Point Defiance, Shed Aquarium. Where's Ben? Omaha. Yeah, there are a lot of people working on the project because it's a big project.
Very cool. Wow. Thank you guys so much.
Thank you, Josie, for your questions. Next question is from Michelle. Michelle, do you mind unmuting?
Hi there. Thank you for your talk. It's really interesting. Are there efforts to collect urchins in quantity, or is it just kind of like a lost cause at this point?
For feeding or just to get them out of the kelp forest?
To get them out of reduce the numbers in the environment.
Yeah, there's a lot of folks who are doing that work. Blanking on who? Reef Check?
Yeah.
Reef Check. I know a few years ago I had the opportunity. They had a control site and a test site. So they let an urchin baron just be an urchin baron, and then we went through and got rid of all the urchins from the test site to see, you know, if we can get rid of urchins, can kelp come back? Because there are other issues, right? There's global warming and the blob, and you know, without the kelp there, can we recruit? And this is just anecdotal, but I was able to see that test site come back and have recruitment. You know, we had an understory, and then the last year I was out there, the actual giant kelp was starting to come back. So I was able to see little bitty giant kelp plants, and I cried underwater. So we know that it works. So there's definitely people who are doing that work, but it's very, very labor intensive. We were out there every year because the urchins just keep coming back. You know, that kelp is tasty. They just want to eat it. So
Thank you.
yeah.
Thanks, Michelle, for your question. And we have another question from Paula. Paula, do you mind unmuting?
Yeah, I was noticing in your data when you see different numbers of larvae per milliliter, that at the lowest density, there's a lot more variability in what happens. And that might suggest to me as a cell biologist that you've got a couple different effects going on, like at low numbers, the larvae might be somehow helping each other to settle, and then at higher numbers, it starts becoming more competition. Do you have any thoughts about that? Or can you do, I don't know if you can do more fine tests of density, maybe larger volume containers? You could, you know,
Yeah.
tease it out.
Yeah, no, that's a great insight, Paula. And one thing that we didn't tell you about that data is that there was another treatment layer that we haven't fully dug into yet, which is the diet trial that Riah was doing. So they were also getting different diets, and we will hopefully very soon be able to tease out what the diet effect was there as well.
Yeah.
Oh,
We
wonderful.
had more jars at 1 larvae per mil, I believe. There were more replicants within that
Yes.
treatment.
Yeah, and
Yeah.
I think like, yeah, we're gonna be able to test it again because that was a wild spread.
Yeah.
And we're really
Yeah.
curious to what was going on there.
Yeah, you might want to talk to some people who've worked in the biofarm industry because we spent a lot of time, trying to figure out how to grow mammalian cells in tissue culture. And they're very shear sensitive, but they need the oxygenation and the mixing. And just a lot of engineering and science has been done to what's called stirred tank reactors. And, you might be able to get some help from people in that neck of the industry.
Yeah, that's a
Yeah,
great
that's
idea.
Yeah.
What were the reactors called again?
Stirred tank reactors, like if you're going to make a monoclonal antibody, they started with 1 milliliter or 5 milliliters, and now they're up to like 12,000 liter reactors. And just scaling up stirring is very challenging
Yeah.
because the surface area to the volume ratio is changing.
Yeah.
And so, I had an engineer ask me what the wall shear rate was, and I had to say, the what? So,
Yeah, thank you for that.
Thank you.
Thank you, Paula. Our next question is from Dan. Dan, do you mind unmuting and asking your question?
Sure. Hi, thanks so much for your presentation. Super interesting, and I think you're on the right track as far as I'm concerned. But I'm just wondering, do you know how old the sunflower stars have to be before they can start eating urchins?
Oh, our newly settled stars were eating baby urchins immediately.
Oh, really?
Tiny, tiny ones. So even if you have
Wow.
a newly recruited Pycnopodia, that helps with newly recruited urchins, right? Because they're coming in waves.
Interesting.
So they'll eat them basically a day or two after settlement.
Wow. Fantastic. Great. Thanks so much for your presentation.
Yeah.
Thank you, Dan. We have a question from Amber that I'm going to read out loud. So Amber says, "You Rule, thank you for the work you do and sharing today. Do you have insight into the genetic variation of Pycnopodia at CIS compared to those observed in the wild in California in the past year and compared to historic pre-blob populations?"
Yeah, so to clarify, we currently don't have any adults here at Cal Academy. So there's very few adults in aquariums in California at all. There's a few at Birch Aquarium, which is where we did the spawn for this particular cohort, and then there's a few at Aquarium of the Pacific. And there's very few, so at less than 5, I think, at this point.
Yeah, I think Heal the Bay has one. I think there might be
6 of them.
5 or 6, yeah.
So the aquarium held diversity is quite low, mostly because there's so few of them. So that is a huge bottleneck and challenge for us right now, and that's another part of why we're really interested in trying to find more of these adults in the wild, and are trying to think about, if and when we find these adults in the wild, how can we harvest their gametes and start spawning them as well. And in particular, because the ones that are currently still out in the ocean most likely have already been exposed to this disease and are the few that survived it. And so those are the ones that, for me, I'm very interested in being able to breed and have hope for having successful reintroductions in the future.
Yeah, and birch houses, all of the animals at the Birch Aquarium have been exposed to seawater their entire lives that they've been at that aquarium. So they've been exposed to wasting, and they're still doing great.
Yeah.
Thank you. Yeah, and Amber was also wondering if people visit California Academy of Sciences, can they see some of your stars?
Yes, so we have 6 of the stars that came from the larvae that settled and we raised in house that are on exhibit now. So if you go down into the aquarium and you find the moon jelly cylinder that's kind of in the middle of the aquarium, take a look to your left, find the grunt sculpins and the spiny lump suckers, and then there's a really small tank just to the right of them, and you can see them cruising around in there.
Awesome. Thank you. And then we have a question from Larry. Larry, do you mind unmuting and asking your question? Or I'm also happy to unmute. Let's see. Okay, so Larry's question is, can you discuss plans for considering reintroduction to the wild? This would presumably start with exposure to raw seawater. I assume they're now being raised in sterile seawater right now.
Yeah, there's a lot of plans in discussion. They're correct that I don't know if I would call our seawater sterile, but it is artificial seawater. So it's not natural ocean water that the stars here are exposed to. Although, as Riah mentioned, in San Diego at Birch Aquarium, the parents of these stars were being exposed to natural seawater, although not raw. Probably it was probably being.
And they've got raw seawater.
Oh, do they? Okay. Yeah, great. So yes, there are discussions of do we start with exposures to raw seawater in aquariums, or do we start with putting some animals out in cages, out actually in the ocean as a trial? So all of that is under consideration right now, which is really exciting.
Gotcha. Thank you. And it looks like our next question is going to come from Vicky. Vicky, do you mind unmuting and asking your question?
Yeah, my camera's not working, so no video, but this is really kind of following along the heels of the question that was just asked. And I think it's probably getting a little bit ahead of the game, but what do you anticipate would be some of the factors in determining when and how you would release the pycnos?
So a lot of this will come into play when we're talking to California Fish and Wildlife, but things we're looking at are size. What size do we want to release at? Where do we want to release? Is it better to release in a kelp forest that is still established, or is it better to release directly onto an urchin barren? Time of year, how close it is to freshwater outlets. So we've seen up north that the younger stars tend to prefer eelgrass beds and basically areas near river mouths. So that's all things that we'll have to consider. And the numbers that we want to release to, if we want to release a population that can maintain itself, and of course a genetically diverse population. So we need to make sure, the stars that we have at CAS right now are not going to be released. They're all direct siblings, and having those breed out in the wild would be disastrous. So they'll stay within aquarium collections.
Thank you.
Thanks, Vicky, for your question. I think our next question is, we just have some comments in the chat. One person, Marilyn, says, thanks for your great work. Hope you're staying under the radar of Doge. We have a comment from Ursula saying, thank you so much for your work and presenting the information. I live on the Monterey coast and I can see the damage to the kelp beds. And then, question from Michelle. Michelle, do you want to unmute and ask?
Yeah, it's a little noisy here. I don't know if you can hear me, but have you done any experiments on water temperature rise and how that, if it contributes to the wasting disease?
It's on our list to try. Yeah, we haven't yet, but it's definitely salinity tests, temperature tests, pH tests are all things that we're hoping to look at.
And in the wild, there have been studies done by other folks that indicated that wasting disease did coincide with a warm water event, and that it seems like the two are quite likely linked.
Thank you.
Thanks, Michelle. We have a question from Sylvie. And anyone else, please enter your questions into the chat if you have them, and we'll get to as many as we can. But a question from Sylvie, yeah.
Yeah, I just wondered, what is the lifespan of the sea stars?
It's a great
I mean,
question.
if they're not eaten by their sisters and brothers.
Yeah, so I don't know a day. No, that's a great question. We don't quite have an answer to it. We used to think 25 years, but the birch aquarium animals were taken into human care in the 80s when they were full grown, and they're still going. So 60 years, 70, we don't know.
So cool. Thank you, Sylvie. Any other questions from anyone? Please go ahead and enter them in the chat. I had one question actually. When you had mentioned earlier using chemical cues for both the stars and the urchins to help them, like to help trigger their spawning, is that sort of cue present in a lot of different marine creatures, and is that used a lot in research? It seems like such a cool technique, and I was just wondering how widely that's used.
Don't know the answer to that.
I think it's pretty commonly used. I think we're pretty lucky in that echinoderms, of which sea stars and urchins are part of, have actually been studied for quite a long time. So if any of you have taken an intro bio lab in college anytime recently, like there's often students like spawn urchins in a lab in order to learn about gametogenesis and things like that. So that type of work has been done for a long time. I think it's much more recent to be broadening out into these other very different non-urchin species. But yeah, I think it's there's quite a bit of research, at least in some species.
That's so cool. Thank you. And I got a question through direct message asking, how old do the sea stars need to be in order to reproduce?
Great question. So that's going to depend on what temperature they're living in and how much food they have, but generally 3 to 5 years.
Gotcha. Cool. Question from Vicky. Vicky, do you want to unmute and ask your question?
Sure. I just was looking up what are the predators for pycnos, and the one thing they mentioned was sea otters. And I know there's a little bit up here in Northern California conversation about trying to reintroduce sea otters. Is that a concern in terms of your project?
I think so.
Room enough for everyone?
Yeah, I think
Yeah.
so.
And the otters, they can eat plenty of other things too, right?
Yeah.
And pycnos,
I would imagine a pycno would not be its first choice.
yeah, pycnos can also release a pretty devastating toxin when they're stressed.
Cool.
So otters can eat them and they like the gametes, but there's other easier things for them to eat.
Urchins.
Yeah, please eat the urchins.
Thank you.
Thank you. This was awesome. I think it looks like there's no more questions, so maybe we can wrap up around now. But just want to thank you, Elora, and you, Riah, so much for joining today and sharing your expertise, sharing your experience and what you've been up to at Cal Academy and all of your work in conserving the species. And yeah, it was just terrific to learn from you all. But maybe everyone, if you don't mind, just like unmuting, we can all say thank you. I can count down 3, 2, 1, and then we can all just unmute and say thank you. So, 3, 2, 1.
Thank you.
Thank you, thank you, thank you.
Bye for all your work.
Yeah.
Wonderful.
Awesome. Thank you all. I hope you all have a great rest of your day, and this recording will be sent out within a week. Thank you all for joining, and if you're interested in more events like this, please consider joining as a Bay Nature member and check out the article that this was written by Guananí all about this. Yeah, thank you all so much for being here, and great to see you all. Thank you, Elora and Riah.
Thank you so
all.
much.
Thank you for your interest.